After getting the subtracted values, next step is we have to take the square root of each value. 110 minus 110 is 0. 0 square is 0. 110 minus 130 means 3. 3 square is 9. And similarly 2 square 4. 1 square 1. And after getting the square values, we have to add all the square values and take the square root according to the SD equation. Then we will get a value of 1.7 and it is our SD value. So now we got the SD value. From the SD value and the mean value, we have to calculate 1 SD and 2 SD. So already we know our mean value is 110. We have to add the SD value 1.7. So we will get 111.7. That is the plus 1 SD. For getting minus 1 SD, we have to subtract my 1.7 from 110. Next is plus 2 SD. So from the 1 SD, we have to add again SD 1.7. Then we will get a value of 112.7. Similarly, for getting minus 2 SD, we have to subtract 111.7 minus 1.7. That is our minus 2 SD value. Similarly, we can find out plus 3 SD and minus 3 SD, so on. So, this is the method of how calculating SD plus 1 SD plus 2 SD. This is the graphical representation of plus or minus 1 SD and plus or minus 2 SD. Next is we have to apply Westgard rule whether our test is acceptable or rejected. So first value is or first rule is 1 2s rule. That means one control value outside plus or minus 2sd. This is known as 1 2s rule. That is one value outside of plus or minus 2sd. Usually we know for a normal Levy-Jennings chart or the quality control, all the values should be within plus or minus 2sd. Then only it is acceptable. This is an exception. If one control value is outside plus or minus 2sd also, it is acceptable. That is the Westgard rule, first one. Next is the second rule, that is 1 3s, 1 2s rule. That is one control value outside of plus or minus 3sd. That is the 1 3s rule. It is definitely a rejection criteria and this is because of random error. Already we discussed how to solve the random error in the previous slide. And next rule is 2 2s rule. 2 2s rule means 2 consecutive days control value outside of plus or minus 2sd. That is 2 consecutive days we got the value which is outside of plus or minus 2sd. And this is definitely because of the systemic error. And next rule is R4S rule. That is plus 2sd we got one day and the next day minus 2sd. This is because of random error. And next rule is 4-1s rule. That means 4 consecutive values outside 1sd on the same side. This is because of systemic error. 4-1s rule indicates that 4 consecutive values outside 1sd on the same side. And next or last rule is 10x rule. 10x rule means 10 consecutive values on the same side of the mean. So these are the Westgard rules. Next is another important, equally important to SD. That is the term coefficient of variation. The percentage of coefficient variation takes into consideration the magnitude of the overall result. And the equation for coefficient of variation is SD divided by mean value into 100. The percentage CV expresses the SD as a percentage of the mean. Lower the CV, better the precision. Lower the SD, more accurate the result. So we can compare these two test results. For potassium, the calculated CV value is 2% and for sodium, calculated percentage is 1.4%. So from this, It is clearly indicating that sodium has the better CV, and in this case it is performing better than potassium because CV value is lower than potassium. So sodium has a better coefficient of variation. Why use a percentage coefficient variation for analysis of results? It allows comparison of precision on different sets of data with the different magnitudes. So CV is primarily used to compare the precision on different sets of data. So making the precision, we are giving more concentration to the coefficient of variation. Next is little bit about the criteria of a good quality control material. First one is it should be available in sufficient quantity, it should be stable, keep in small volumes, and its concentration should vary minimally. Their composition should not vary from vial to vial, and control material should be tested like the test sample. So these are the criteria of a good quality materials. It should be available in sufficient quantity, should be stable, always allocate in small volumes and keep in correct temperature in refrigerator, and its concentration should not vary. Their composition should not vary from vial to vial, and also temperature is also very important while storing the quality control materials, and it also tested similar manner of a test sample, routine test sample. Next is about the classification of quality control. Quality control is broadly classified into internal quality control and external quality control. Internal quality control is designed to check that a laboratory will produce the same result or outcome if the test or procedure is done on different occasions. So this is mainly used for detecting laboratory variations or by different technicians. Accuracy is to ensure day-to-day consistency of an analytical process. So it's very clear that is internal quality control is used to check the laboratory variations. So accuracy is applied to all work processes and to every test done in the lab, and every day it is must to do internal quality control. And accuracy procedures identify problems immediately. We have to rectify it and again reload the test samples. Next is about the external quality control. External quality assessment or proficiency testing allows a comparison of laboratories' test procedure to other laboratories across the world. So, internal quality control is used to assess the variation inside the lab and in between the lab staffs, whereas external quality control is, from the name indicating, it is used to compare the lab to an external reference lab. So comparison can be made to the reference laboratory and result of the individual laboratory are compared. So this is the pictorial representation of an external quality result. If a laboratory performs 1 to 25 parameters per day, then one level QC one time should use. If the parameters are in between 25 to 75, then two level QC use, and 75 or more, it should be used two level QC two times to give a better result. Next is little bit about the accreditation agencies. First one is NQAS, National Quality Assurance Standards, and ISO, International Organization for Standardization, NABS, National Accreditation Board for Hospitals and Healthcare Providers, NABL, National Accreditation Board for Testing and Calibrating the Laboratories. So these are the external accreditation programs. So I hope this is very clear, and quality is everyone's responsibility. So in this class, we detailedly discussed about the quality control, what is quality assurance, and about the classification of errors, pre-analytical, analytical and post-analytical errors, and the classification of quality control, quality chart, and the dress code rule, and the term specificity, what is sensitivity, accuracy, linearity. So study all these things. So if you like this video, please subscribe and share my channel. Thank you.